Allan Doctor of WashU, St. Louis.
All right, thank you, and very much appreciate the opportunity to talk with you this morning about Erythromer, which is a novel nanoscale biosynthetic artificial red cell. So there's been an unmet need for some time for a blood substitute to enable transfusion where stored blood is unavailable or undesirable. Unfortunately, all prior blood substitutes have failed due to two key flaws: poor oxygen delivery to tissue, despite being able to improve oxygen content in blood, and a problem with vasoconstriction, where the blood substitutes cause vasospasm, preventing traversal of normal red cells to tissue. Because of major improvements in synthetic chemistry and nanomedicine, we've been able to encapsulate normal human hemoglobin with a suite of small molecules that encode wetware, if you will, that coordinate normal or imitative behavior for the particle that simulates red blood cells. Our design is comprised of the following components. The particle shell either sequesters or releases a small molecule that modulates oxygen affinity in connection with blood pH, so that in the lung, oxygen affinity is high, and in tissue, oxygen affinity is low. The particle shell is designed to develop a selective diffusion barrier to nitric oxide, a vasodilator molecule, that prevents sequestration and consumption and the vasospasm that plagued prior blood substitutes. We also include a small molecule that retards iron rusting so that hemoglobin doesn't oxidize during use of the blood substitute. And the blood substitute itself is designed to be freeze-dried, so it can be stored for extended periods at ambient temperature and reconstituted easily with water at the point of care. A molecule that was designed to imitate the normal components of a red cell membrane are put in a vessel with the payload components. It self-assembles into a toroid about 1/50th the size of a red blood cell, and at the end of synthesis, the surface is cross-linked to improve particle stability and to develop the selective diffusion barrier for nitric oxide. We exploited a genetically engineered mouse model to design a novel efficacy test. We used a reporter molecule called HIF, or hypoxia-inducible factor, that glows, is genetically engineered to glow when it's stabilized. This detects the presence of hypoxia. Here you can see a mouse that is glowing and anemic. Here is a mouse that's not glowing, who's been resuscitated with the blood substitute. 70% of his blood volume has been replaced with the artificial red cells, which when clear, the glowing returns, demonstrating not only effective capture, but release to tissue and the absence of vasospasm. We envision several opportunities for Erythromer use outside the resuscitation of wounded soldiers or trauma victims, possibly for use for short-term in the operating room, or to deliver oxygen past vascular obstruction, or to preserve viability of explanted organs prior to transplant. We have a highly choreographed plan to bring this to human trials, which we anticipate will take about 8 to 10 years. I want to also acknowledge the vast team of colleagues that helped with this, a synthetic chemist at University of Illinois, Urbana-Champaign, and a specialist in mouse models of transfusion at the University of Toronto. Thank you for your attention.
Thank you very much, Allan.